It is my great pleasure today to introduce Dr. Sam Golden. He's an assistant professor, right?
Assistant professor.
Okay, assistant professor as
don't let the beard fool you. I'm still young.
Yeah. Probably soon to be, I'm sure soon to be associate professor in the Department of Biological Structure at the University of Washington. Thanks, Sam, for joining us today.
Thanks for having me. I'm looking forward to this.
Yeah, and so I just wanted to start by mentioning that Sam and Marco Venero have recently put together a workshop which was hosted by European Behavioral Pharmacology Society, the sort of sponsor or associated entity of psychopharmacology in Rome last year on social behavior. And he's currently organizing, I think also with Marco, a special issue on social behavior. And so they are currently still recruiting submissions. So if you have some behavioral neuroscience to do with social behavior, get in touch with Sam and he will take a look at your submission. So welcome, Sam.
Yeah.
And so we're going to talk today starting on a paper that you published, a review paper you published a couple of years ago in Psychopharmacology by Nastasia Goodwin, Simon Nilsson, and yourself. It's called Rage Against the Machine, Advancing the Study of Aggression Ethology via Machine Learning. And I thought this was a really interesting paper, and I wanted to talk to you today about not only your research on aggression and social behavior in general, but also some of the machine learning approaches you've been using to try to quantify behavior in a more efficient way and make this more accessible to behavioral neuroscientists that may be part of the audience of psychopharmacology. So to get started, you are interested obviously in the neurobiology of aggression. So can you just tell us why aggression is important to study?
Yeah, I think the history of studying aggression as a field, as a neuroscience field, has really been fascinating. Obviously, I came in after it's been pretty far along. This is a field that had some beautiful work in the 40s and 50s and 60s that really heavily influenced our own work, especially when it comes to this idea of appetitive or rewarding aggression. I can talk about that in a little bit. And then, in the 80s and 90s, it sort of switched from behavior to genetics. And that led to, I think, a pretty strong decrease in the interest overall in the study of aggression until more recently with the wonderful work with optogenetics and chemogenetics. There's been this sort of just complete renaissance in the field, because now that we have tools that allow us to look at these complex behaviors at the speed at which they occur, the speed at which they're encoded, people have become much more excited again at the basic phenomenon and understanding it. So you see this historically, there's a lot of interest in aggression, then there was a lot of not interest in aggression, and now we've got this resurgence again. So it's a really exciting time. Personally, for me, I got interested in studying aggression after being in the addiction field a little bit. And I became especially interested in this phenomenon because it's just so fundamentally conserved and naturalistic. A lot of times when I was taught about addiction, people would say, oh, look at how this is, something that just at the most basic level hijacks the reward circuitry, right? And then I'm sitting here looking at social behavior and I'm thinking, well, that was here before drugs. It does the same thing. And I was really curious in how that works. And especially when it comes to the transition between adaptive social behavior and maladaptive social behavior. So, aggression itself is a fundamentally adaptive response. We all have it. We all use it in all different parts of our day-to-day life. If we don't, then we're seldom as successful as those that do. So the really interesting thing in my mind is first understanding that basic phenomenon, but then what happens when it transitions to a compulsive or addiction-like state, when it's no longer guided by adaptive choice. So for example, you don't fight normally if you know you're going to lose. Mice are no different. If they know it's a bad call, they back off, except in some cases when we see them transition to this compulsive or maladaptive state, which is often facilitated by things like alcohol or other interactions with pharmacoactive substances. And so those interactions have always just been super interesting to me. I always thought, well, I'd like to know more about it. And I got very lucky that in graduate school, my PI at the time, Scott Russo, was willing to take a risk and allow me to completely branch off course halfway through my PhD and started working on this new idea of aggression motivation. And then as a postdoc, I was also by Yavin Shaham given just absolute leeway in a lab that does, at the time, nothing with social behavior to pursue this new direction. And I think it was really a lot of luck by having mentors that were willing to allow me to take a risk, while still certainly under their watch, which led to this opportunity to really develop a research program based upon understanding the molecular and circuit mechanisms, looking at why some animals like to fight and others find it equally aversive. Interesting.
It's kind of a testament of the benefits of good mentorship. I think we've many of us have experienced. So yeah, that's really great. And then as far as the actual doing of this research, can you tell us a little bit about some of the specific challenges that come up when you're trying to quantify social behaviors like aggression?
Yeah. Well, there's so many. Where to start? So the first thing I think that one of the reasons that social behavior in general is not utilized as fully as it could be is that it is really hard to annotate and annotate in a non-subjective and consistent manner. And we see this all the time. If I bring a bunch of undergrads, and we have a lot of undergrads in the lab, they bring a lot of life to the lab, and I appreciate that. But when we bring them in and we ask them to look at a video that's already been annotated for social behavior, so we know it's a gold standard video, and it's 10 minutes long, so it's not even that long, right? And we train them and we ask them to annotate it. We see something all the time, which we call the Reddit effect, which is after 3 to 5 minutes, you can sort of tell when the undergrad starts surfing Reddit, as much as maybe paying attention to the behavior, and that's no fault of their own. It is nothing more monotonous and brain melting than having to sit there and hand score hours and hours, if not hundreds of hours of video. And that's exactly what I did as a graduate student. And when I started my own lab, a couple of things happened. First, we were a brand new lab, and second, COVID happened, and we couldn't do anything. And I had a fantastic postdoc at the time who's since gone on to an industry startup position. And we were sitting there, and I'm going, well, we can't run any animals. And but I have many videos that you can hand score. And he looked at me, dead in the eyes and goes, "I'm not doing that." And I go, "I understand. So what other approaches are there?" And just at this time, the first preprints had come out for things like Deep Lab Cut, and at the time Leap, and also Deep Pose Kit. And these were all pose estimation architectures that allow you to use computer vision approaches to track specific features in a given frame. So basically the nose of a mouse, the left and right paw of a mouse. And it gives you the X and Y coordinate in every single frame. And we thought, well, this tool is pretty phenomenal. Maybe we can use it by taking it to the next step of taking that type of pose estimation data and building upon a lot of other open source tools that have been doing this in their own unique ways for many years now, adapt it specifically for complex social behavior and to classify and predict using supervised initially, but now also unsupervised approaches when animals are performing something that normally would have to manually annotate. And so that problem, right, of just it's so boring and so subjective and so easy to have poor inter-rater reliability, and the very very lucky timing of these other pose estimation open source tools coming out at the same time, and the very unlucky reality of COVID happening led to this confluence of events where we said we can overcome these problems using these computer vision and machine learning approaches. And that was sort of the genesis of a pipeline we've been developing in the lab called SIMBA, which stands for Simple Behavioral Analysis. We joke about it. This is Nastasha Goodwin came up with this acronym, and we immediately loved it, not because the mice's or the rat's behavior was simple, but because we as behavioral neuroscientists trying to adapt these tools are simple, and we needed it to work for us, not computationally trained, hopefully not computationally poor, but certainly not computationally trained behavioral neuroscientists. How can we adapt this in such a way where we could use it as well? And initially it was just a bunch of scripts in Python, and then we would show them to our collaborators, and they'd be like, "Oh, we really want to do that." And we're like, "Yeah, here's the scripts." And then they'd be like, their eyes would roll back, and they'd be like, "We don't want to do this." And then we'd end up spending as much time walking them through using the scripts as actually generating data. And so we said, well, maybe we can, again, look at some of the open source tools that are already out there, like Java from Kristen Branson's lab at Genelia, and also the great work at that time that was happening with these pose estimation tools to put a GUI on it. And we said, well, let's make it accessible. So we started working on a graphical user interface. And from there, it just sort of, we got the preprint out as fast as we could. And it just sort of spun much more out of control than we thought. We just actually passed 450,000 downloads, which blows my mind.
Wow.
And over 100 citations on the preprint, and many workshops every couple of months. We do 2 or 3 workshops. And it's just become a tool that people are using in a way that we did not anticipate, and which we're not really set up to support as a small academic lab, but we're doing our best. But it's been really exciting. And that all started because of these very basic problems. So the other major issues that come up with machine learning right now in pose estimation are if your mice look the same. So everyone wants to do multi-animal, multi-agent tracking, right? And multi-agent pose estimation, behavioral classification, and the problems that are still exist, although there's certainly many fantastic groups working on this right now, is the problem of recording in naturalistic environments where occlusions often occur. So you can imagine if you put something as simple as an igloo in your home cage, and 2 similar looking mice go under the igloo, as they do, and then 2 similar looking mice are absent from video for a couple of seconds and they shoot out in opposite directions. And comically, the question is, well, which one is which? And if your biology has directionality, where one animal is your experimental mouse and the other one is your non-experimental, those ID switches can cause a huge amount of problems. And so right now there's some great work from LabCut, and also Leap now became Sleep, where they've introduced ways to try to prevent or correct ID switching in similar looking animals. We still find that the best approach is to use dissimilar looking animals. So we spent a lot of time, for example, when we work with C57s, which are generally black coded, we now work with white coded intruders that are C57s that are simply missing a gene for pigment. And we do a lot of behavioral controls to ensure that coat color is not something that's unduly influencing our other social behaviors. But once that's done, we really like this approach because it's so simple. And it all comes back to simplicity for us, which is what can we do that gets it to be as easy for people to reproduce and take advantage of. And while there are these amazing tools coming for multi-animal tracking, at the end of the day, the simplest thing for me when I'm just looking at a video is still if they're a different color. And so we've sort of gone in that direction. But the occlusions are a horrible problem, right? If you lose the animals, they get occluded. How do we fix that? And there's a lot of work on that problem right now ongoing.
Yeah, I noticed in your paper you mentioned that one of the approaches, other than just having 2 different color animals, which obviously I agree simplicity is usually the best strategy, is sort of like tagging the animals, like RFIDs and stuff like that. Like, is that something that you see as really promising going forward, or something that's not?
Yeah, there's a fantastic group that put together something called Live Mouse Tracker, and it came out in like 2018, and it combines RFID tracking with continuous video capture. And what it does is that when the animals are together and they cannot be disambiguated, as soon as they separate far enough apart that their RFIDs can be individually tagged again, which means whenever they go over a different antenna separate, that can then allow to reattribute the correct ID. And of course, paired with the motion capture or the video capture, that's really, you can always pop back to the right animal no matter what. And it's a fantastic approach. So these approaches are really good. The downside to them, and this is like the classic struggle, right, is generalizability versus specificity. So what I mean by that is we developed Simba from the mindset that there's a lot of labs out there that have a lot of old video, and a lot of video looks really crappy and really different, and it's like suboptimal, and like, can we get something from it still? Versus building a setup from the get-go that is always going to be identical, always will have optimal parameters for that specific application. And they, both of these have a lot of pros and cons to their approaches. Simba is never going, for example, any type of generalizable or supervised classification scheme is never going to perform as well as an optimized one in that specific environment. But the optimized one in that specific environment might only be applicable to a single open field, which maybe doesn't capture all of the context that we would want to use normally. And so, for the users out there who are thinking of adapting machine learning approaches, you really have to unfortunately, dive into the literature. We tried to make it a little easier with this review and some of the more recent reviews, but dive into the literature, look at the dozens and dozens and dozens of open source tools that are currently available that span this spectrum of, like, we build a box, use the box, to very generalizable, but you have to work a little harder to get it to be, to have really good fidelity. And you have to pick what works best for your application. And there certainly are reasons to go one way or the other. So for us, we really like the generalizable aspect because we do all sorts of different behaviors. Everything from operant behavior within chambers to open field to condition place preference. And we like to be able to classify social behaviors within all of these different types of arenas and lighting conditions and video capture conditions without having to have a single setup for each of them. And so for us, that's been very good. For other people who are looking at open field behavior as their primary readout, I would say, no, you got to go with a specialized setup, because in the long run, if that's what you're doing, you'll get better and more consistent predictions that way. So it's really, you got to pick your poison, as I think is always.
It's always the struggle in these kinds of things, of course. And I'm very comfortable with that trade-off of, obviously generalizability is extremely important, and especially if you're going to be, translating this or using this in other species and very different sorts of video situations and stuff like that. There's a huge advantage to that. But as far as the specificity part of it, I'm comfortable with the idea of being not 100% accurate in terms of like implementing this in my own research, let's say. Certainly, as you mentioned, the Reddit effect is real, and the undergrad scored stuff is also not 100% accurate, and so that's just kind of the nature of the beast. What sort of I worry about, I wouldn't say keeps me up at night, but something like that, is that these catastrophic failures that can sometimes occur with these, especially with black box approaches and things where you're just letting the algorithm tell you what it thinks it sees, and sometimes I think you used a nice example. You can use ML approaches to black box ML approaches to like tell the difference between a wolf and a husky, but also actually it's really detecting if there's snow in the background, something irrelevant that is correlated with the thing you care about. And these things kind of do worry me. So could you speak a little bit about kind of that trade-off and this idea of like supervision and unsupervised algorithms and things along those lines?
Yeah, absolutely. We spend a lot of time thinking about that. In fact, I'd say over the last year, we've pivoted really from developing Simba as a supervised classification tool, because it does that, it's fine, but more to introducing mechanics for explainability and transparency in the predictions that are made. And the reality is right now we are in the wild west of these approaches, right? I sort of liken it to when next-gen sequencing was just starting in the early 90s, and people were generating these insanely rich data sets that, we look back now at how they were analyzed and we go, maybe we put a little too much in it. And that's where we are right now. Right now with computer vision and machine learning for behavior, we can generate data on a scale that would have been, just absolutely unthinkable. So Nastasia right now is working on a data set between just two groups of animals that have about 20 hours of video each for this experiment, and she can crank through that in a day or two using these automated approaches, which would have been her whole PhD in a past era. And
similar to my PhD, actually.
What? Sorry.
Similar to how my PhD was spent just hours in front of a video.
Exactly, and I would wish that upon nobody. So there's a lot of personal motivation to make sure this works. But like you're saying, so if we're putting that much data through, how do we ensure that it's accurate? Because we're not going back and putting eyes on every single frame. It's not possible. And so what we've really been working towards most recently is introducing tools from the computer sciences world, where there are 30 or 40 years ahead of where we are now, and trying to adapt them to our behavioral purposes. And one that we're really happy with currently, and that we introduced earlier this year in a current opinion and neurobiology paper, is called SHAP, or for Shapley additive values. And this is basically an approach that allows us to take all of the features that make up a classifier. So right, let's take a step back. We'll break it down real simply. So you've got those XY coordinates of the nose, of the tail, of the left and right flank. And what we're really doing when we're saying we're predicting a behavior is we're taking all of those XY coordinates across different time windows and extracting all of the relationships between them that we can think of that are useful. Orientation, velocity, size, shape, distances, different time windows, all of these, until we get a list of maybe 500, 600, 700 features that are derived from that pose estimation between however many animals we're interested in. Once we have those features, we use them to create what we call a random forest. It's just one of many algorithms that are available that help a computer make a decision between if a target or if a behavior is present or absent in a given frame based on all those relationships from that pose estimation over different rolling windows. That's, yeah, maybe it does sound complicated, but that's it. That's all it is. And the nice thing about supervised approaches rather than unsupervised approaches is that we do have these analytical tools available that allow us to identify how much any one feature is contributing to the overall predictive strength of that classifier. And we use one of them. We've been a proponent of using one of these, which are these SHAP values. So SHAP is just a game theory approach. It's really straightforward. Let's say you've got, we'll make it easy. Let's say you've got 10 features in your classifier, and you want to know how important any given feature is. Well, what SHAP allows you to do is almost a brute strength approach, where it will randomly pull one of those features out of your classifier and then recalculate how good of a classifier it is off of a known baseline data set. And it then does that for each of the features in your data, in your classifier, changing the order at which they're pulled until it brute forces through all of these permutations. And then it attributes to each one of these features a value. And the sum of all of those values equals if a prediction, 1, if a prediction is happening versus 0, if a prediction is not happening. And now we have metrics for each one of these features that we can use to better understand why that decision was made. And so we use this for all sorts of reasons. This explainability and it isn't just for the biology. Let's say you're studying aggression because it's awesome and you want to study aggression, and I'm studying aggression. Now you're studying it and you're doing it in C57s and I'm doing it in CD1. So inbred versus outbred, very different behavioral spectrums, but we're studying aggression. So now you're going to say in your, in your Nature paper, in the methods section, you're going to say, and then we scored when the animals attacked. And that's sort of the operational definition. But I know from experience that how a two C57s fight is massively different than how two CD1s fight. And now the question is, how can we reconcile across labs and across research groups what these actual behaviors are? Well, if you generate an attack classifier and I generate an attack classifier and we extract the SHAP values for the features that we're using, and there's some nice mathematical reasons that we can directly compare them, even if our pose estimation is a little different. The long of it is, or the short of it is then I can exactly ask, well, what are the key features that are driving why you're calling that aggression? Oh, well, for you, in your C57s that are fighting, it's the distance between their faces over this time window and the shape of the resident's body. Whereas in my mice, it's all based on the intruder's behavior or whatever. The point is now we don't have to fall back on sort of some, verbal definition of these different behaviors. We can fall back directly on quantified metrics that we can share the same way you would share, the RRID of the antibody that you used. You would, in the future, we hope, be able to share SHAP or other, I don't really care what people use, so some other metric that defines what these behaviors are. With this, that's at the very basic level. Between labs, we might be able to better reproduce and standardize what's going on. But on the flip side of that, let's say, well, listen, for my science paper, I don't want everything to be the same. I have a very nuanced behavior that I've discovered. The way my animal is grasping for that is critical for engaging the manifolds or whatever. Whatever the point is that someone wants to do. That very specific behavior, well, it still needs to be defined in a way that other people can accurately understand it. And again, whether it's for standardization of things we're all doing or for better defining a very specialized behavior, the incorporations of these types of transparency and explainability metrics will make it much easier to transfer these between labs, or at least understand them between labs. And then, of course, we get into the fun stuff, right? Neural recording. when we start looking at how specific behaviors are associated with specific neuronal ensembles. Well, now that we can actually map them, quantifiably break down the different components in any given frame of behavior that are actually occurring and leading to a behavioral prediction with that neural recording data. And so it starts to get a lot more interesting and a lot more robust. And so those are sort of the directions that we hope by increasing and getting away from these black box approaches, we'll be able to better understand not just the behavior, but the neurobiology driving the behavior. Now, there is a whole field of computer science dedicated to this type of work. And we just scrape the surface and try to pull the most accessible and easy to understand components of that into our thinking. But this is the exact same approach. The people that have automated driving cars, they need to understand, why is it a cone and not a person? These are the same things. And so, we're not discovering those approaches. We're simply identifying the approaches that seem to be working well already, and how can we incorporate them into behavioral neuroscience. And so SHAP is one we're really a proponent of, and other labs have already adapted it and published with this approach. And so we think it might work. It might work, but time will tell, obviously.
I think this is a really valuable approach, if nothing else, just for transparency across labs and comparability and stuff. And so one thing that occurs to me, though, with this approach is that I think you gave a really interesting example. You have a mouse, I have a mouse. Our mice both aggress, and we're both studying aggression. But my mouse aggresses in a sort of physically different way for whatever reason than your mouse does. But we would still, as a field, want to call both of those things aggression. This is aggression in my strain, that's aggression in your strain. And so this then, I totally understand the value then of taking, the concept of aggression and trying to map it onto a series of XY coordinates and then have all the transparency and everything that we just talked about. But it does sort of start with the concept that there is a thing called aggression that we want to categorize in a certain way and then compare across labs. So in a way, this is like the supervision part. This is the human input part. It's like, what are we trying to define is defined by us. And to me, this is like a little bit of a tension in the field. It seems to me, as a naive outside observer, is that, one of the huge values of this kinds of approaches is that maybe the machine or the cloud server or whatever it is that I'm using to analyze this stuff is going to tell me something brand new that I never expected before, that my human brain would never know about a mouse that is actually really important to a mouse, but I'm putting my human sort of interpretations on top of that. And yet, there's also seems like you're saying some of the most effective and useful approaches sort of start with a presupposition of there is a thing we're trying to define. So could you just speak to that sort of those two different things that we're trying to do here kind of with these approaches?
Do we have 3 or 4 hours that we can go? I know. So you're totally right. So there's this give and take between supervised and unsupervised, identifying the unknown behavioral repertoire versus automating something that I'd otherwise spend 1,000 hours of man or woman power time on, right? And they're two totally different things. So when I study aggression, at the end of the day, I, based on 100 years of literature, feel very confident that we know when aggression occurs. Like I would gladly be wrong about this, but I don't think a computer is going to identify aggression that we have missed. Now, that said, what I cannot do, and what no one in my lab can do, is identify the different types of attack clusters that are occurring during these aggression bouts. And unsupervised approaches, for example, can do that. And so there are 2 ways that people can look at this. Purely unsupervised, where you just take all your data and you throw it in. This works, look at the data labs work, just absolutely mind-blowing. It's so beautiful. Fantastic when you have an individual mouse in an individual cage, and you want to do some type of manipulation, and you want to understand all of the constituent components of these sequences of behavior that are occurring. And these unsupervised approaches do very consistently identify unknown repertoires or sequences. I believe the data lab calls these syllables, that make up the language of behavior, and that works really well. The problem with this is that many of these approaches, and I'm looking forward to whoever solves this, it'll make my day, require sort of an egocentric view for the mouse. What does that mean? Well, the mouse is a one mouse in a cage, and they can align everything off of that mouse's sort of center line. Does it go to the left? Does it go to the right? Does it scrunch up? Does it stretch out? Right? And all of the syllables, or just we'll call them motifs, are based off of sort of that egocentric view. But when you incorporate a second animal for complex social behavior into these types of events, you got to throw that out the window, because now it takes two to tango and it's not egocentric anymore. And so to the best of my knowledge, or at least not published yet, no one has identified an unsupervised approach for mice that are engaged in robust, complex social behavior that can sort of in an unsupervised manner take advantage of these amazing advances that are already out there and apply them in that setting. And that probably is going to come out tomorrow in like Nature Methods, but today that's what we got. So our approach, and this is an approach that other labs have now adapted successfully, is to say, well, we know what attack behavior looks like. We know what aggression looks like. So let's take all of our aggressive bouts, extract those video clips, and only run unsupervised analysis on that attack behavior. And now what we've done is we've sort of really winnowed down all the noise that would be a part of our analysis, and we're just looking at that attack. And when we do that and we perform unsupervised analysis on multiple mice only during these epochs of different supervised behaviors, we can begin to identify some very consistent type of attack behaviors. And so this is something we're hoping to submit very soon, where we look at under two different contexts, both sex as a biological variable and outbred mice, so looking at male and female outbred mice and what their attack structures look like, but also looking at male mice in two different environmental contexts, literally where the arenas are different shapes, and how that influences the different types of attacks that we see, which we would never, ever be able to see by eye. I mean, it's just too much, too fast, too subjective. But that now, using unsupervised approaches, we can very clearly reproducibly identify. And so, we're still, like, it's still a wild west, like I said, we're still identifying what are the, not even the best, but what are the most, what works, right? Let's just say what works to identify these types of novel or unique behavioral sequences in these already supervised spaces. And so that seems to be working pretty well for us right now. And so, for example, when this paper comes out, we've identified 3 attack types in females and 2 attack types in male outbred mice that are very consistent. And then when we change the environment so that the animals have more flexibility in like what they can do because they have more space or less space, this is very obvious. But what we see is that the structure of the attack types that they use are completely dependent upon the environment that they're in. And this is awesome and amazing, but it's also horrifying, because that means now, let's say I'm doing unsupervised analysis in my social defeat experiment that uses a long, wide box, and you're doing unsupervised in your home cage, and she's doing unsupervised in her operant chamber. And then we're saying, oh, look at all these clusters. Well, are these truly unique clusters, or are these contextually dependent behaviors that we're identifying? So it becomes, I mean, it's science. It's a very interesting question, but not a very easy question. And so that's sort of, I think, the downside and also upside of the direction we're going with all of these unsupervised and supervised approaches, is that with this degree of ability to dissect behavior, it's very easy to start to lose the trees, right, in the forest. And so I think we have to be very aware of that, which is why I like supervised as our starting point, because we found the tree, now we're cutting down the tree and figuring out how it's made. And so that, I think, at least for us, a non-computational lab is a good way to start.
Absolutely, yeah, and I think there's room, obviously, for all these approaches in where we're going here as a field, and I really do think that these kinds of integrating the fields of AI and its many applications to our very specific type of application is totally tractable. It just requires people like you really to kind of help bridge that gap and kind of speak both languages and things, and easier said than done. But ultimately, as you said, all these challenges that emerge out of this, the way I look at it is now they become tractable problems. Now we can actually compare apples to apples and see what is.
I will say though, the key is not people like me. The key are the amazing postdocs and graduate students that are doing this work. And then I am simply the test. If I get it, if they can explain it to me in an intuitive way where I'm like, oh yeah, that does make sense, then that means it's probably worth putting into the package in the pipeline. And so, you know, really the work being done here is being done by people like Simon Nilsson, Nastasha Goodwin, JJ Chun. These are all people who are or were in the lab that have really, you know, they're the ones that did all of this work. I just sit back and go, oh my God, that's so cool. It's so useful. And like, show me the next thing. Now we have the next generation in the lab that are working on this. Kevin Schneider and Carly Todds, Giovanna Navarrete, and all of them are really the people who are not classically trained, right? And they take on all this extra time and effort to learn this stuff on top of how they were trained as behavioral neuroscientists. And it's very, I think that this is one of the reasons it's been a successful approach, is because the perspective is from the user rather than from the computer science side. And sometimes we have some very frustrating, but ultimately very hilarious conversations with the computer science side, you know, and they go, well, I don't understand why that's not working. And I go, well, have you ever done a surgery on a mouse and tried to do what you're suggesting with us? And like, and they go, oh, wait, that's right, there's a mouse involved in this. And so, you know, it's really the combination, but ultimately it's the amazing people in the lab that are doing this work. I just get to, you know, I just get to sit back and look at it.
Yeah, we're very lucky. Yeah, and ultimately it's going to be, you know, people talking to each other that have their own expertise. This is the cool thing about neurosciences. You know, we have all kinds of scientists that are approaching this hard problem of how the brain makes behavior and et cetera. And it's going to take, you know, a village, so to speak.
Yeah, and thankfully the NIH is starting to acknowledge that, and they're starting to provide some funding mechanisms for dissemination of these tools. Not nearly enough to support them in a real way, but enough that now, for example, there's a great workshop that's going to be run, that is actually, it just happened, at a JAX that involves some of the leaders in this field, where trainees can go and learn how to use these open source tools. And that's like an R25 mechanism that was funded by Kumar Vivek and a few others. And I think that those like types of approaches, like that's sort of the issue where we are now. There's so many tools coming out. There's so many approaches. Obviously, everyone's heard of deep lab cut. Everyone's heard of sleep, but have you heard of dance? You know, no. Well, okay. Well, that's how you do this in 3D. And this, it's such a rapidly advancing field and such a like a vibrant open source community with so many people building and putting things out there that it's very easy to get lost. You know, where do I start? Great question. Yeah. Not an easy question. So
not easy, but I think your review actually is a great place for people to get started. To, you know, some excellent tables in there that lay out some of the pluses and minuses of these various different groups approaches and things. And you know, this is I think it's really a service to the field in the sense that you know, people can kind of take it as a starting place, see what is the application that's going to work best for their preparation and so on. And you know, together, I think all of us, myself excluded, of course, here, will then advance this field, which is absolutely where behavioral neuroscience is going, in my opinion. And I think we should wrap it up there. But this has been really a fascinating conversation, Dr. Golden. And you know, I think, like I said, your paper is going to be a really influential one, and I'm really excited to see some of the next steps that you have in this on this research program. You and your very talented trainees.
Well, thank you so much for giving me the opportunity to share what they're up to and some of the things that are coming out of the lab and just how we think about it. And you know, I'll be the first to say as a closing thought, we might be very wrong, and we would love and welcome feedback as we go, because yeah, this is sort of a new direction for the field, and it is a collaboration between some really different interdisciplinary approaches. And so there are some things that we find work really well, and then there's some things that we think work really well that don't, and there's some things that don't work for us that work really well for other people. And what we're hoping is that the open source nature of this movement will lead to more communication and more abundant communication as this all gets worked out, mostly, you know, through GitHub preprints and sort of Twitter non-traditional publishing methods, which are much faster and I think much more adaptive to this type of work. And so, yeah, join the conversation. If you're watching this and you've got a thought or a comment or a question, you know, we always love to hear it. And there's a lot of people out there who would love to hear it as well.
Absolutely. And one of the formats you can make your voice heard, in addition to contacting Dr. Golden, is submitting to his special issue of Psychopharmacology on social behavior. So if you have something to add to this, ML-wise or otherwise, you know, feel free to send that on in. Yeah. And thank you again, Dr. Golden. This has been a great conversation, and I look forward to seeing what you're up to next.
All right.
Right.
Thank you. Oh, you're muted.
Sorry about that.